Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Rawal from Dr. APJ Abdul Kalam Government College, Silvassa, UT of Dadra and Nagar Haveli. Today we will discuss about yield of energy from glucose under the paper Carbohydrate Metabolism. So students, you learn about the glycolysis, you learn about the TCA cycle. So the question must come in your mind, what is the outcome of glucose metabolism? So over here we will learn glucose as a key metabolite in plants, animals and many organisms. We try to understand how much yield of energy from glucose we are getting. We will touch the aspects of the cellular respiration and biochemistry behind ATP yield from glucose. Energy may be most simply defined as the capacity to do work or to cause particular changes. Thus, all physical and chemical processes are the result of the application or movement of energy. Cells must efficiently transfer energy from their energy generating or trapping apparatus to the system actually carrying out the work. As we learn in the electron transport chain, at the end there is a generation of ATP, and this actually the ATP that converted into the useful work. That is, cell must have a practical form of an energy currency. In living organism, the major currency is adenosine triphosphate, that is ATP. When ATP breaks down to adenosine diphosphate and orthophosphate, energy is made available for useful work. So it is actually the breakdown of ATP that releases the energy and that can be converted into useful work. And that's why we say the ATP is the currency of cell. Later, energy from photosynthesis, aerobic respiration, anaerobic respiration and fermentation is used to synthesize ATP from ADP and orthophosphate. Since ATP is generated within the mitochondria, that's why we say it as a powerhouse of a cell. But in the case of the prokaryotes, this electron transport chain lies in the plasma membrane, while in the case of the eukaryotes, it lies within the inner membrane of mitochondria. Metabolism, it is the sum total of all chemical reaction occurs in the cell. It can be divided into catabolism and anabolism. In catabolism, molecules are reduced in complexity and free energy is made available. Energy also can be derived from sunlight by the process called photosynthesis and the oxidation of inorganic nutrients. Anabolism involves the use of this free energy to increase the complexity of the molecules for the biosynthesis purpose. During catabolism, nutrients are funneled into a few common pathways for more efficient use of enzyme. A few pathways processes a wide variety of the nutrients. The tricarboxylic acid cycle, that is a TCA cycle, is the final pathway for the aerobic oxidation of a nutrient to carbon dioxide. The majority of energy released in catabolism is generated by the movement of electrons from electron transport carrier with more negative reduction potential to ones with more positive reduction potential. Thus, aerobic respiration is much more efficient than anaerobic catabolism. A wide variety of electron acceptors can be used in catabolism. endogenous organic molecules, that is a fermentation, oxygen, aerobic respiration, and oxidize exogenous inorganic and organic molecules other than the oxygen, that is a part of anaerobic respiration. Furthermore, reduced inorganic molecule as well as organic molecule can serve as an electron donor for electron transport and ATP synthesis. Microbial catabolism is unique in the diversity of nutrients and mechanism employed to make energy available. While in the case of the eukaryotes, in photosynthesis, trapped light energy boosts electron to more negative reduction potential or higher energy levels. These energized electrons are then used to make ATP and NADPH or NADH during electron transport. Proton motive force is generated by oxidative reactions and photosynthesis. It is used to power the production of ATP and other processes such as transport and motility. The maximum ATP yield in eukaryotes from glycolysis, the TCA cycle and the electron transport can readily calculated. The conversion of a glucose to 2-pyruvate molecule during glycolysis gives a net gain of 2 ATP and 2 NADH. Why I say a net gain of 2 ATP? Because 2 ATPs are being consumed and 4 ATP is produced. So net is 2. Because each NADH can yield a maximum of 3 ATPs during electron transport and oxidative phosphorylation, the total aerobic yield from the glycolytic pathway is 8 ATP molecules. But over here we need to keep in mind the transport of this NADH also to mitochondria expends 1 ATP. Under anaerobic conditions, when the NADH is not oxidized by the electron transport chain, only 2 ATPs will be generated during the degradation of glucose to pyruvate if the condition is anaerobic. Let's look at the concept map of this module. In this module, our target is to find out the yield of energy from one molecule of glucose. Glucose undergoes glycolysis, and then after the pyruvate that is formed will convert it into acetyl coenzyme A, and that acetyl coenzyme A enters into the TCA cycle. So from this entire process, the glucose is getting converted into carbon dioxide and water. The ultimately target over here is to find out the energy that is released from the one molecule of glucose. So over here we need to concentrate on the preparatory phase of the glycolysis, where the 2 ATP is actually consumed. Another phase of the glycolysis, that is the glycolysis payoff phase. Over here 2 coenzyme yield, that is 2 NADH we are getting, and ultimately 4 ATP is formed by the substrate level phosphorylation. So we need to take account of this. At the same time, oxidative decarboxylation step of the pyruvate will give 2 NADH. Similarly, from the Krebs cycle, we have a substrate level phosphorylation, oxidative phosphorylation of NADH, oxidative phosphorylation from the 2-FADH2, and then after we need to come on yield from the single molecule of glucose. So first of all, we need to go through the pathways of the glycolysis briefly, and the oxidative decarboxylation step and the Krebs cycle, and then after, picture will be clear in your mind how much ATP is formed from the substrate level phosphorylation and how much ATP we are getting through electron transport chain by NADH and FADH2. So we need to count how much NADH is generated from glycolysis, from oxidative decarboxylation, from Krebs cycle. At the same time, how much FADH2 has been formed. We need to take account of the energy expense. Glucose basically is an aldohexose which act as a key metabolite in many plants, animals, and microorganisms. In some microorganisms, it is the only source of energy. It is a good fuel due to its rich potential energy. Breakdown of glucose is the major source of cellular energy. Suppose if we eat starch containing food, then ultimately that polysaccharide is converted into monosaccharide, and that monosaccharide, that is nothing but the glucose, will enter into the glycolysis. Let's look at the cellular respiration. The complete oxidative breakdown of glucose to carbon dioxide and water, that is C6H12O6 plus 6O2, will give 6 carbon dioxide and 6 molecule of water. During this respiration process, a large amount of free energy, around 686 kilocalorie per mole, is released in the reaction. One glucose molecule gives 38 ATP molecules. Over here, my friends, we haven't accounted the expenses. So, the one glucose molecule gives 38 ATP molecules during the cellular respiration. But practically, some ATPs are utilized for moving pyruvates, for moving phosphates and ADP into the mitochondria. That has not been accounted over here. We need to account that, and then after we will come to the yield of energy from the glucose. In the screen, we are observing the ATP yield in a typical eukaryotic cell. The glucose undergoes glycolysis, where 2 ATP is consumed, 4 ATP is produced, 2 NADH is produced. Then it undergoes oxidative decarboxylation, where 2 NADH is produced. That forms the acetyl coenzyme A, which ultimately enters into the Krebs cycle that releases 2 ATPs, 6 NADH and 2 FADH2. So students, we are exploring over here how much energy we will get from one molecule of glucose. Obviously, the glucose first enter into the glycolysis. It results into the 2 molecules of pyruvates. That pyruvates then get converted into acetyl coenzyme A, and that enters into the TCA cycle. The glycolysis is a series of reactions that takes place in the cytoplasm of all prokaryotes and eukaryotes. As you know that glycolysis is neither aerobic nor anaerobic. Glycolysis converts one molecule of glucose into 2 molecules of pyruvate. These molecules of pyruvate are then converted to acetyl coenzyme A. This acetyl coenzyme A is then ready for the entry into the citric acid cycle. 2 ATP molecules are needed for early reactions in the glycolytic pathways. That reactions are called energy consuming reactions. But 4 ATPs are generated later in this the same pathway, which gives a net yield of 2 ATP molecule of a glucose degraded. Now, the citric acid cycle, as you know that the 2 pyruvates that is formed will get converted into the 2 molecule of acetyl coenzyme A, and these 2 molecules of the acetyl coenzyme A then enters into the citric acid cycle. The citric acid cycle oxidizes pyruvate formed during the glycolytic breakdown of the glucose to carbon dioxide and water, with the concomitant production of energy. Acetyl coenzyme A from fatty acid breakdown and amino acid degradation products are also oxidized. In addition, the cycle has a role in producing precursors for the biosynthetic pathways. The cycle forms the central part of 3-step processes, which oxidize organic fuel molecules into carbon dioxide with the concomitant production of ATP. During the TCA cycle, we discussed already that it produces the many precursors for the other pathways, for the amino acids, for the fatty acids. We discussed the same during the anapyruretic reactions. This diagram explains the process of glycolysis. Although some energy is obtained from the breakdown of glucose to pyruvate by the pathway which is known as glycolysis. Glycolysis involves the conversion of glucose to glucose 6-phosphate. This glucose 6-phosphate is getting converted into fructose 6-phosphate. This fructose 6-phosphate will converted into fructose 1,6-biphosphate by the enzyme phosphofructokinase. Now, this fructose 1,6-biphosphate will splits from 6-carbon compound to 2 molecule of 3-carbon compound, that is a glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This glyceraldehyde 3-phosphate, as you have learned in the process of the glycolysis, so I will not go much in detail over here, my dear students. This glyceraldehyde 3-phosphate is ultimately converted into phosphoenol pyruvate, and this phosphoenol pyruvate will ultimately converted into pyruvate. The first part, that is a preparatory phase, where glucose is getting converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, consumes the 2 ATP, while the last part, that is a payoff phase of the glycolysis, will gives the 4 ATP. Now my friends, let's look into the steps of the glycolysis. We already discussed briefly the same. The glucose is getting converted into glucose 6-phosphate by the enzyme hexokinase. Glucose 6-phosphate to fructose 6-phosphate by the enzyme phosphoglucoisomerase. Fructose 6-phosphate is phosphorylated by ATP to form fructose 1,6-biphosphate and ADP by the enzyme phosphofructokinase. Over here, 1 molecule of ATP is consumed. In the first step also, the 1 ATP molecule has been consumed. Aldolase enzyme splits the fructose 1,6-biphosphate, that is a 6-carbon molecule compound, into 2 3-carbon molecule, that is a glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The different steps of this glycolysis has been shown in the previous slides. The glyceraldehyde 3-phosphate and dihydroxyacetone phosphate has been formed from the glucose. Now this glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. The newly created high energy phosphate bond of 1,3-bisphosphoglycerate is now used to synthesize ATP by the enzyme phosphoglycerate kinase. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. The enolase enzyme catalyzes the dehydration of 2-phosphoglycerate to form phosphoenol pyruvate. Pyruvate kinase catalyzes the physiological irreversible transfer of the phosphoryl group from the PEP, that is a phosphoenol pyruvate, to ADP to form ATP and pyruvate. So in the glycolysis, the fate of glucose is pyruvate. 6-carbon glucose is getting converted into 2 molecules of 3-carbon containing pyruvate. In this, 4 ATPs are produced by the process substrate level phosphorylation and 2 ATPs are consumed. So the goal of glycolysis is breakdown of glucose and conversion of the glucose into 3 carbon containing 2 molecules of the pyruvates. All organisms performs the glycolysis, whether it is prokaryotes, eukaryotes, whether it is bacteria, plants or animals. The glycolysis occurs in the cytoplasm of the cell. As I mentioned earlier, do remember this, glycolysis produces 4 ATPs and 2 NADH, but it utilizes 2 ATPs in the process. So, ultimately, we are getting net 2 ATP and 2 NADH from this process. There are two distinct methods by which cells synthesize the ATPs. As you know, my dear friends, the oxidative phosphorylation and substrate level phosphorylation. The oxidative phosphorylation involves the electron transport chain, the transfer of electron and the coupling with that, the synthesis of ATP. In oxidative phosphorylation, the generation of ATP is linked to the oxidation of NADH and FADH2, which occurs via the electron transport chain. The electron that is donated by this reduced NADH and FADH2 releases the electron. That electron transports through the electron carriers, which are namely complex 1, 2, 3 and 4. This transfer of electrons generates the gradient. That proton gradient actually drives the synthesis of ATP across the inner mitochondrial membrane through the ATP synthetase enzyme. The 2 ATP synthetic reaction in glycolysis, which is catalyzed by phosphoglycerate kinase and pyruvate kinase, involves the direct transfer of phosphate from sugar phosphate intermediates to ADP. These reactions are examples of substrate level phosphorylation. Why it is called substrate level phosphorylation? Because over here the phosphate group is transferred directly from the intermediates, that is a sugar phosphate intermediates, to ADP and results into the formation of ATP, and that's why it is called what? A substrate level phosphorylation. We learned the glycolysis, and the fate of glycolysis is glucose is converted into 2 molecules of pyruvate. Now this pyruvate will undergo oxidative decarboxylation step. The reaction shows the oxidative decarboxylation of pyruvate, which goes further in the Krebs cycle. This oxidative decarboxylation step is very important because it converts the pyruvates into the acetyl coenzyme A, and actually this acetyl coenzyme A, it enters into the TCA cycle. Over here, 1 molecule of NADH is formed. So dear friends, glucose is converted into 2 pyruvate molecules. Now these 2 pyruvate molecules undergo decarboxylation steps to generate what? 2 NADH. A major source of energy is a glucose, which is converted by glycolysis into pyruvates. The pyruvate dehydrogenase, it's a complex of 3 enzymes and 5 coenzymes. Then oxidize this pyruvate using NAD, which is reduced to NADH, and ultimately it forms acetyl coenzyme A and carbon dioxide. Do remember this, 1 molecule of carbon dioxide is removed from the pyruvate from this. So pyruvate actually is 3 carbon contained. Now it becomes 2 carbon. Since the reaction involves both an oxidation and a loss of carbon dioxide, this process is called oxidative decarboxylation. Now we already have a look on the glycolysis oxidative decarboxylation step. Now let's move to the next part where this acetyl coenzyme A enters, that is a citric acid cycle. The citric acid cycle, also known as TCA, that is a tricarboxylic acid cycle or a Krebs cycle, is used to oxidize the pyruvate formed during the glycolytic breakdown of glucose into carbon dioxide and water. It also oxidizes the acetyl coenzyme A arise from the fatty acid degradation and amino acid degradation products. In addition, the cycle provides precursor for many biosynthetic pathways. The citric acid cycle operates in the mitochondria of the eukaryotes and in the cytosol of prokaryotes. Succinate dehydrogenase, the only membrane-bound enzyme in the citric acid cycle, is embedded in the inner mitochondrial membrane in eukaryotes, while in the plasma membrane of the prokaryotes. The cycle forms the central part of a three-step process which oxidizes organic fuel molecules to carbon dioxide with the release of ATP. Now let's look into the steps of the citric acid cycle. Citrate is formed from the irreversible condensation of acetyl coenzyme A in oxaloacetate, which is catalyzed by the citrate synthase. Citrate is converted to isocitrate by an isomerization reaction catalyzed by aconitase. This is actually a two-step reaction during which cis-aconitase is formed as an intermediate. It is the cis-aconitate which gives the enzyme its name. Isocitrate is oxidized to alpha-ketoglutarate and carbon dioxide by isocitrate dehydrogenase. Actually, in this step, one molecule of carbon dioxide is removed. So now the 6-carbon isocitrate is converted into 5-carbon alpha-ketoglutarate. Remember this, 1 carbon from the pyruvate is removed from the oxidative decarboxylation step. Over here, 2nd carbon has been removed. Now, this mitochondrial enzymes requires NADE plus, which is reduced to NADH. Alpha-ketoglutarate is oxidized to succinyl coenzyme A and carbon dioxide by the alpha-ketoglutarate dehydrogenase complex. So again, now this 5-carbon alpha-ketoglutarate is converted into 4-carbon succinyl coenzyme A by release of carbon dioxide. So total all the 6 carbon, all the 3 carbons of the pyruvate has been removed. First in the decarboxylation step from the pyruvate to acetyl coenzyme A, then in the isocitrate dehydrogenation step, and now in this alpha-ketoglutarate dehydrogenase complex steps. Like pyruvate dehydrogenase, this is a complex of 3 enzymes and uses NAD plus as a cofactor. Succinyl-CoA is converted into succinate by a succinyl-CoA synthetase. The reaction uses the energy released by cleavage of the succinyl coenzyme A bond to synthesize either GTP, mainly that is in the animal, or ATP, that is exclusively in plants, from inorganic phosphate, and respectively either GDP or ATP. Succinate is oxidized to fumarate by succinate dehydrogenase. Over here, FAD is tightly bound to the enzyme and is reduced to produce FADH2. Fumarate is converted to malate by fumarase. This is a hydration reaction requires the addition of a water molecule. Malate is oxidized to oxaloacetate by malate dehydrogenase. NAD plus is again required by the enzyme as a cofactor to accept the free pair of electron and produce the NADH. Each turn of the cycle gives 1 molecule of GTP, 3 molecules of NADH and 1 molecule of FADH. The citric acid cycle completes the oxidation of glucose to 6 molecules of carbon dioxide. 4 molecules of ATP are obtained directly from glucose molecule, 2 from glycolysis and 2 from the citric acid cycle, 1 for each molecule of pyruvate. In addition, 10 molecules of NADH, 2 from the glycolysis, 2 from the conversion of pyruvate to acetyl coenzyme A, and 6 from the citric acid molecule, and 2 molecules of FADH2 are formed. So total 10 NADH and 2 FADH. The remaining energy derived from the breakdown of glucose comes from the reoxidation of NADH and FADH2, with their electrons being transferred through the electron transport chain to reduce oxygen to water. So FADH2 NADH that comes out of this glycolysis, TCA cycle, oxidative decarboxylation steps, then undergoes electron transport chain. They donates the electrons to the electron transport chain. Electron transport chain operates in the inner membrane of the mitochondria in eukaryotes. Electron transport and oxidative phosphorylation occurs in the inner membrane of the mitochondria. This process reoxidizes the NADH and FADH2 that arise from the citric acid cycle, which is again located in the mitochondrial matrix, and glycolysis, which occurs in the cytoplasm, and fatty acid oxidation, which again located in the mitochondrial matrix, and traps the energy released as ATP. Oxidative phosphorylation is by far the major source of ATP in the cell. In prokaryotes, the component of electron transport and oxidative phosphorylation are located in the plasma membrane. Electrons are transferred from NADH to oxygen along the electron transport chain, which is also called the respiratory chain. NADH passes the electrons to the complex 1. Electron transport is normally tightly coupled to the ATP synthesis. Electron transferred to complex 1 will eventually transfer to other complexes, and finally it ends to the terminal electron acceptor, that is in the aerobic respiration is oxygen. Electron transport is normally tightly coupled with ATP synthesis. Electrons do not flow through the electron transport chain to oxygen unless ADP is simultaneously phosphorylated to ATP. Just remember the inhibitors of the electron transport chain, where the uncouplers can uncouple these two processes, which ultimately ends into the release of energy in the form of heat. During oxidative phosphorylation, the electrons of NADH and FADH2 combines with the oxygen, and the energy released from the process drives the synthesis of ATP from ADP. The transfer of electrons from NADH to oxygen releases a large amount of energy, 52.5 kilocalorie per mole for each pair of electron transferred, so that this energy can be harvested in usable form. The process takes place gradually by the passage of electrons through a series of carriers which constitute the electron transport chain. The components of the electron transport chain are located in the inner mitochondrial membrane of eukaryotic cell. In aerobic bacteria, which uses a comparable system, components of the electron transport chain are located in the plasma membrane. In either case, the transfer of electron from NADH to oxygen yields sufficient energy to drive the synthesis of 3 molecules of ATP. Electrons from FADH2 enters the electron transport chain at a lower energy level, so the transfer to oxygen yields less usable free energy, only 2 ATP molecules. So students, now let's come to the net energy yield from the aerobic respiration of a glucose. Glucose is converted into pyruvate. At that time 2 ATP is consumed and the 4 ATP is produced. So the net yield from the glucose by the substrate level phosphorylation is 2 ATP. This pyruvate is getting converted by oxidative decarboxylation into acetyl coenzyme. This acetyl coenzyme enters into the Krebs cycle, which ultimately gives 2 ATP. So the net ATP yield by substrate level phosphorylation from glycolysis and from the Krebs cycle is 4 ATP. Most of the ATP that we are getting, that is through electron transport phosphorylation, that is around 32 ATPs. My dear friends, you might have remembered we got 10 NADH molecules. Each NADH produced in the glycolysis is worth a 2 ATP. The NADH that is produced actually having a 3 ATP value, but the transfer of this NADH from cytoplasm to the mitochondria expends 1 ATP. So there is a net gain of 2 ATP for each NADH produced in glycolysis. Each NADH produced in the conversion of pyruvate to acetyl coenzyme and Krebs cycle is worth 3 ATPs. So that is around 8. So 8 3s are 24. FADH gives 2 ATPs. So 2 2s are 4. 24 plus 4, 28. And the 4 ATPs that we are getting from that. So net energy production, that is 36 ATP. So students, in this lecture we try to explore how much energy that we are getting from one molecule of glucose. We touch the aspects of the glycolysis, tricarboxylic acid cycle, that is TCA cycle, glycolysis and citric acid cycle yields a total 4 molecule of adenosine triphosphate directly from each glucose molecule. That is again the part of substrate level phosphorylation. In addition, the 10 molecules of NADH, that is 2 from glycolysis, 2 from the conversion of pyruvate to acetyl coenzyme and 6 from the citric acid cycle, and 2 molecules of FADH2 are formed. This FADH2 and NADH delivers the electrons to the electron transport chain, and that transfer of electrons through the electron transport chain will generate a proton gradient, which generates the proton motive force, which drives the synthesis of ATP. So this remaining energy derived from the breakdown of glucose comes from the reoxidation of NADH and FADH2, with their electrons being transferred through the electron transport chain and reduce oxygen to water, that is H2O. Theoretically, a total of 38 ATPs are formed from complete oxidation of glucose, but practically 36 ATPs are produced due to the loss of some ATP in transport. In this lecture, we learn about the catabolism of glucose. We try to explore the energetics aspects of the glycolysis and Krebs cycle, how ATPs are generated during the electron transport chain, and yield of ATP from a glucose. Thank you very much.